Plants produce a large variety of secondary compounds that contain a phenol group or a hydroxyl functional group on an aromatic ring. These substances are classified as phenolic compounds or phenolics. Plant phenolics are a chemically heterogeneous group of nearly 10,000 individual compounds. Some are soluble only in organic solvents, some are water-soluble, and others are large, insoluble polymers. In keeping with their chemical diversity, phenolics play a variety of roles in the plant. Many serve as defenses against herbivores and pathogens. Others function in mechanical support, in attracting pollinators and fruit dispersers, in absorbing harmful ultraviolet radiation, or in reducing the growth of nearby competing plants. Plant phenolics are synthesized by several different routes and thus constitute a heterogeneous group from a metabolic point of view. Phenylalanine is an intermediate in the biosynthesis of most plant phenolics. Two basic pathways are involved, the shikimic acid pathway and the malonic acid pathway. The shikimic acid pathway participates in the biosynthesis of most plant phenolics. The malonic acid pathway, although an important source of phenolic secondary products in fungi and bacteria, is of less significance in higher plants. Lignin is a complex phenolic polymer or highly branched polymer of phenylpropanoid groups, mainly derived from the three monolignol, coumaryl, coniferyl, and synapyl alcohols. As an important component of secondary cell walls in vascular plants, lignin is the second most abundant plant-derived organic substance after cellulose. Lignin fills the space in the cell wall between cellulose, hemicellulose, and pectin components, especially in vascular and support tissues including xylem, plaquette, vessel element, and sclerate cells. Lignin is covalently linked to hemicellulose and therefore closely links different plant polysaccharides, conferring mechanical strength to the cell wall and by extension the plant as a whole. Its physical toughness deters feeding by herbivorous animals, and its chemical durability makes it relatively indigestible to herbivores and insect pathogens. Lignifications block the growth of pathogens and are a frequent response to infection or wounding. A second category of plant phenolic polymers with defensive properties besides lignin is the tannins. They are general toxins that can reduce the growth and survival of many herbivores when added to their diet. In addition, tannins act as feeding repellents to a great variety of animals. Mammals such as cattle, deer, and apes characteristically avoid plants or parts of plants with high tannin content. Unripe fruits, for instance, frequently have high tannins level, which deter feeding on their fruits until their seeds are mature enough for dispersal. Herbivores that habitually feed on tannin-rich plant material appear to possess some interesting adaptations to remove tannins from their digestive systems. Plant tannins also serve as defenses against microorganisms. The flavonoids are one of the largest classes of plant phenolics. The basic carbon skeleton of a flavonoid contains 15 carbons arranged in two aromatic rings connected by a 3-carbon bridge. Flavonoids are classified primarily on the basis of the degree of oxidations of the 3-carbon bridge. The four major groups of flavonoids are anthocyanins, flavones, flavonols, and isoflavones. Different types of flavonoids perform very different functions in the plant, including pigmentation and defense. The color pigments of plants provide visual cues that help to attract pollinators and seed dispersers. These pigments are of two principal types, carotenoids and flavonoids. Carotenoids, as discussed earlier, are yellow, orange, and red terpenoid compounds that also serve as accessory pigments in photosynthesis. The flavonoids include a wide range of colored substances. The most widespread group of pigmented flavonoids is the anthocyanins, which are responsible for most of the red, pink, purple, and blue colors observed in flowers and fruits. Anthocyanin color is influenced by many factors, including the number of hydroxyl and methoxyl groups in ring B of the anthocyanidin, the presence of aromatic acids esterified to the main skeleton, and the pH of the cell vacuole in which the anthocyanins are stored. Anthocyanins may also exist in supramolecular complexes along with chelated metal ions and flavone copigments. Two other groups of flavonoids found in flowers are flavones and flavonols. These flavonoids generally absorb light at shorter wavelengths than do anthocyanins, so they are not visible to the human eye. However, insects such as bees, which see farther into the ultraviolet range of the spectrum than humans do, may respond to flavones and flavonols as visual attractant cues. Isoflavonoids, which are a group of flavonoids found mostly in legumes, have several different biological activities. Some, such as rotenone, can be used effectively as insecticides, pesticides such as rat poison, and pesticides or fish poison. Other isoflavones have anti-estrogenic effects. For example, sheep grazing on clover rich in isoflavonoids often suffer from infertility. The ring system of isoflavones has a three-dimensional structure similar to that of steroids, allowing these substances to bind to estrogen receptors. Isoflavones have also become known for their role as phytoalexins, antimicrobial compounds synthesized in response to bacterial or fungal infection that help limit the spread of the invading pathogen.